Good afternoon, all of you. Today's topic of my presentation is the construction of the 5x174 gene mediated lysis system for the generation of Salmonella-Enteritidis ghost and its evaluation of its vaccine potential against the avian salmonellosis. The bacterial food poisoning is a major worldwide health problem, and Salmonella remain among the leading sources of the foodborne illness. Among the Salmonella serovars, the Salmonella-Enteritidis is a non-host adapter organism and is capable of infecting variety of host animals, including human being. And the infection of human is acquired through the consumption of the contaminated poultry meat and contaminated poultry eggs. This is some statistical figures in Korea. The prevalence rate of the Salmonella-Enteritidis infection is about 18.58% through the fruit products. And the worldwide figures are Salmonella-Enteritidis is responsible for 93.8 million illnesses and 1,500 deaths worldwide each year. So definitely, for control of this zoonotic pathogen, the control measures have been targeted by using the chemotherapeutics or by using the live, killed or subunit vaccines. Here we represent some of the newer, it's not actually newer approach in the history of science, but this was recently implemented for the Salmonella-Enteritidis vaccine, the generation of the genetically inactivated vaccine by means of the bacterial ghost technology. As summarized in the before presentation, I will not go through the details what is meant by bacterial host and all, but in this process we use the genetically controlled system for the generation of Salmonella-Enteritidis ghost, and we use the bacteriophage 5x174 lysis gene E, which codes for the 91 amino acid polypeptide, which assembles in the inner and outer membrane of the gram-negative bacteria, leading to the formation of hole, and the cytoplasmic content gets out of the hole because of the difference in the osmotic pressure of the media and the cytoplasm. And as the outer membrane structures or the cell surface envelope of the bacterial ghost is not getting affected by any chemical denaturation process, the antigenic determinants are still intact, and these antigenic envelopes we can use as a vaccine for protection against the infectious disease. So this is the basic architecture of the genetic inactivation system, what we call it as a ghost cassette. This ghost cassette has a 5x174 lysis gene E, which is controlled by the temperature sensitive repressor system. This is the CI857 and lambda PR promoter. This regulatory system also originates from the bacteriophage lambda. Now the mechanism of the control of the lysis gene E. At the temperature up to 30 degrees centigrade, the CI857 repressor protein forms a dimer and which binds the operator sites of the lambda PR promoter and which blocks the sitting down of the RNA polymerase for the initiation of the transcription. However, if we shift the temperature from 30 to above 40 or for example 42, that time this higher temperature causes the denaturation of the dimer of the CI857 protein, and it cannot bind the operator region of the lambda PR, and it subsequently initiates the transcription of the downstream cloned gene E, which leads to the production of protein E and subsequently the bacterial ghost formation. Taking this idea in mind, in order to have the bacterial ghost, we cloned our ghost cassette comprised of the gene E and the regulatory system in the very simple plasmid is a TEZ vector from Promega, which had the ampicillin resistance genes for the stable maintenance in the host system. So we transform wild type Salmonella-Enteritidis with this TEZ vector containing ghost cassette, and the transformed strain we inoculated in the LB broth, and after growing, after reaching the growth at the mid-logarithmic phase, we shifted the cultures at 42 degrees centigrade, and subsequently we monitored the OD and CFE of the cultures, and after 48 hours we harvested the ghost cells by means of centrifugation. This is the data of the OD and CFU which during the lysis hours at 42 degrees centigrade. If we can monitor the OD, this line represents the OD values of the Salmonella-Enteritidis which doesn't have the ghost cassette, and these are the OD values which from the Salmonella ghost strain, which is transformed with the ghost cassette. We can see that once the temperature was induced, because of the effect of the lysis protein E, the cells started dying and there was no further growth, which is also can be visible in case of the CFU graph. This was our first achievement in the development of the bacterial ghost vaccine, Salmonella ghost vaccine, and that we published in the Infection and Immunity, which gives the proof that this system was successful. Yeah, although our previous system, which had the ampicillin resistance gene, was successful for the generation of bacterial ghost, but the usage of the antibiotic resistance gene is always a problem in the vaccine production. And as we could detect the presence of the DNA, ampicillin resistant gene DNA in the bacterial ghost preparation, we thought we should shift to some other system which will be antibiotic resistant gene free. So we chosen another backbone plasmid which had the metabolic ASD gene for the stable maintenance into the bacterial host set, and we cloned the ghost cassette on this ASD gene containing plasmid, and we formed the new lysis plasmid which called PJHL101. Then we created a ghost strain, Salmonella ghost strain, by means of a balanced lethal host vector system, which we knocked down the ASD gene, the aspartate semialdehyde dehydrogenase gene of the wild type SalmonelloEnteritidis host, and then we transformed this ASD positive ghost plasmid in this strain, and the SalmonelloEnteritidis ghost strain was created because of the balanced lethal host vector system. And this we published in the PLOS one, the antibiotic resistance gene free ghost cassette plasmid. Till now I presented two plasmids which has the ghost cassette, and this was the basic architecture of the ghost cassette. But during our subsequent experiments, we found that this CI857 repressor protein is not able to completely repress the lambda PI promoter, and there was a leaky expression of this lysis gene E even in the absence of induction temperature, which is visible by the presence of the clumps in the culture even at the 28 degrees centigrade. So what we thought, we added the new regulatory system for the stable control of the ghost lysis gene E in such a way that the lysis gene E was located in the sense lambda PI promoter with the CI857 regulator, and the antisense P-arabid promoter with the RX regulator. This P-arabid promoter is active only in the presence of the exogenous addition of the L-arabinose in the culture. So in the presence of the L-arabinose, the leaky transcription of the lysis gene E at normal growth temperature from the sense lambda PI promoter was repressed by the simultaneous transcription event from from P-arabid promoter through the antisense RNA mediated inhibition. This graph shows the OD values with arabinose and without arabinose. In case of the cultures with arabinose, because of the antisense RNA mediated inhibition of the leaky gene expression, the cells grow very steadily. However, in the culture media, which didn't have the arabinose, we could observe the cell death. This is the picture showing the culture with the arabinose and culture without arabinose. Now the question arises, why we stopped this leaky expression of lysis gene E? Because in order to proceed for the vaccine production, for the scaling up of the vaccine production, we need to have the more cell number to proceed for the ghost production. But because of the leaky expression, we could observe the cell death and we could recover less CFU. And because of this regulatory system, now we have the culture which didn't show any cell death at the normal growth temperature, and which is having the definitely higher CFU, around 10 times higher CFU than this. This we published in the Veterinary Microbiology about our tightly regulated bacteriolysis system. Now here is the electron microscopic characterization of the SalmonellAenteritidis ghost. The single pore appeared at the middle or the polar sides of the areas, mostly the areas which are very active in the cell division. And we can see the single pore and the bacterial cells get shrink because it didn't contain any, it doesn't have any cytoplasm in comparison with the intact bacterial cells. And in the transmission electron microscope, we can see that the clear area without cytoplasm. However, the intact vegetative cells were observed in the unlysed cells. Then we tested this bacterial ghost vaccine, the 100% lysed bacterial ghost vaccine, in the chicken model. We immunized the chickens via intramuscular route, subcutaneous, and even the oral route. We could observe, after immunization, we could observe the significant induction of the plasma IgG response in all the immunized groups, and a significant induction of the intestinal SIGA response in all the immunized groups. And even the cell-mediated immune responses were also stimulated, which are shown by the stimulation index in case of the lymphocyte proliferation assay. We further characterized the vaccine, the induction of the cell-mediated immune responses by intramuscularly immunizing chickens, and we monitored the overall population of the peripheral blood CD4 positive, CD8 positive, CD3 positive, CD4 positive, CD3 positive, CD8 positive T cells, and the population of CD4 and CD8 T cells were significantly higher in the immunized groups in comparison with the control group. Then we investigated the gene expression of the cytokines in the in vitro stimulated cultures from the control and immunized groups. We could observe that the Th1 cytokines were strongly stimulated in case of the immunized groups in comparison with the control group, and the IL-10 and IL-6 cytokines were also significantly stimulated in comparison with the non-immunized group. Then we performed the challenge studies. In case of the challenge studies, we challenged the immunized chickens at 4-week post-vaccination, and the challenge dose was 1 × 10⁹ CFU per bird via oral route. At 1 and 2 weeks post-challenge, we sacrificed the chickens for counting the presence of the number of CFU of the challenge strain in the organs. In case of the liver, spleen, and cecal contents, we could observe a significantly lower count of the challenge strain in immunized group in comparison with the non-immunized challenge group. Then the study that we carried out was in younger chickens. As SalmonellaEnteritidis mostly affect the adult laying hens, we carried out the immunization and challenge study in adult layer chickens. We in one group we immunized chickens by giving prime and booster vaccination, and one group was immunized only single time. And when the egg laying was started, that time the chickens were challenged intravenously with the 1 × 10⁷ CFU per bird. And here is the data of the organ bacterial recovery from the liver, spleen, ovarian follicle, and cecal contents. The count of the challenge strain in the liver and spleen was significantly lower than the control group. However, there was a significant difference between the 2-time and 1-time vaccinated group. In case of the spleen also, we could find the significantly lower number of the bacteria in comparison with the control challenge group. Same case was repeated in the ovarian follicles and the cecal contents. And then we observed the pattern of the egg production and egg contamination. In case of the egg contamination pattern for 3 weeks, we could observe that the significantly lower egg contamination rate was observed in immunized group in comparison with the control group. And this explains that although the bacterial ghost vaccine is not capable of complete prevention of the disease, but it is capable of significantly reducing the occurrence of disease. In conclusions, the stable system for production of bacterial ghost was established. The production of bacterial ghost is a relatively simple approach for production of the highly immunogenic, genetically activated vaccines. And the Salmonella ghost is capable of inducing both the cell-mediated and humoral immune responses, and capable of protecting chickens against the Salmonella infection in chickens. The ongoing progress in this project, now we are testing different adjuvants along with the Salmonella-Enteritidis ghost and Salmonella typhimurium ghost and Salmonella gallinarum ghost for getting the more accurate protection against the virulent challenge. In last, I would like to acknowledge my advisor, Dr. Jawale, and our collaborator, Dr. Samun Kim, for their efforts in the project. Thank you.
Any questions from the floor? Okay, thank you very much.
Yeah, thank you.